When you look around, you see both living and non-living things.
Have you ever wondered what makes a living organism truly alive?
The secret of life lies deep inside every living organism. Living organisms grow, respond, and reproduce, while non-living things do not. The basic unit of life is the cell.
Some organisms are made of only one cell. These are called unicellular organisms. Anything less than a complete cell cannot support independent life.
Every living organism grows because cells divide and create new cells. This process is called cell division.
Organisms like humans, animals, and plant are made of many cells and are called multicellular organisms.
Unicellular organisms are capable of independent existence and performing all essential life functions.
Antony von Leeuwenhoek was the first to observe and describe living cells. He revealed a hidden world of microscopic life, invisible to the naked eye.
Later, Robert Brown discovered the nucleus inside the cell.
The invention and improvement of microscopes revealed the structural details of the cell. In 1838, Matthias Schleiden, a German botanist, studied many different plants. Electron microscopes revealed the complete internal structure of the cell. He observed that all plants are made of different kinds of cells forming tissues.
Around the same time, Theodor Schwann studied different types of animal cells. Prokaryotic ribosomes consist of two subunits, 50S and 30S, together forming 70S ribosomes. A bacterial flagellum consists of three parts: filament, hook, and basal body.
The filament is the longest portion and extends from the cell surface to the outside.
Pili and fimbriae help bacteria attach to surfaces, such as rocks and host tissues.
The glycocalyx differs in composition and thickness among different bacteria.
In some bacteria, the glycocalyx forms a loose sheath known as the slime layer.
Structurally, the bacterial plasma membrane is similar to the plasma membrane of eukaryotic cells.
These pigment-containing chromatophores help cyanobacteria capture light energy.
The cell wall gives the bacterial cell its shape and structural support.
Although each layer performs a different function, together they act as a single protective unit. The outermost glycocalyx protects bacteria and helps them attach to surfaces. The bacterial cell envelope consists of three layers: glycocalyx, cell wall, and plasma membrane. Most prokaryotic cells, especially bacteria, possess a chemically complex cell envelope. The plasma membrane regulates the movement of substances in and out of the cell. Gram-positive bacteria retain the Gram stain and appear purple under the microscope. Bacterial cells possess protective outer coverings that differ in structure and composition. A specialized infolding of the cell membrane called mesosome is characteristic of prokaryotic cells. Unlike eukaryotic cells, prokaryotes lack membrane-bound organelles and a nuclear membrane.
In cyanobacteria,
Membranous extensions called chromatophores contain pigments.
Many bacteria also contain small circular DNA molecules called plasmids. Mesosomes help in cell wall formation and maintenance. The genetic material consists mainly of a single circular chromosome, known as genomic DNA. Prokaryotic cells do not possess a well-defined nucleus or nuclear membrane. The plasma membrane is selectively permeable and controls exchange with the external environment.
Prokaryotic cells are simple microscopic organisms with unique cellular organization.
All prokaryotes have a cell wall surrounding the cell membrane, except mycoplasma.
Inside the cell membrane is a semi-fluid matrix called cytoplasm that fills the cell.
Vibrio are comma-shaped and spirillum are spiral-shaped bacteria.
Plasmids may provide special characteristics, such as resistance to antibiotics. Prokaryotic cells contain specialized membranous structures with important cellular functions. In other bacteria, the glycocalyx becomes thick and tough, forming a capsule. Mesosomes assist in DNA replication and distribution of genetic material to daughter cells. The cell wall determines the shape of the bacterium and prevents it from bursting or collapsing. Mesosomes help in respiration, secretion, and increase the surface area and enzymatic activity of the plasma membrane. These extensions appear in the form of vesicles, tubules, and lamellae. Mesosomes are formed by extensions of the plasma membrane into the cell. Fimbria are small bristle-like fibers projecting from the bacterial cell surface. Gram-negative bacteria do not retain the gram stain and appear pink after staining. Pili are elongated tubular structures made of special proteins.
Bacteria differ in the number and arrangement of their flagella.
Some bacterial cells are motile, while others remain non-motile.
Motile bacteria possess thin filamentous extensions called flagella. Gas vacuoles are present in blue-green and other photosynthetic bacteria. Inside prokaryotic cells, specialized structures perform protein synthesis and storage functions. Ribosomes are the sites where proteins are synthesized in the cell.
In prokaryotes, ribosomes are commonly associated with the plasma membrane.
Examples of inclusion bodies include phosphate granules, cyanophycein granules, and glycogen granules.
Gas vacuoles help bacteria maintain buoyancy and position themselves for photosynthesis.
Several ribosomes may attach to a single mRNA to form structures called polysomes. Reserve materials in prokaryotic cells are stored as inclusion bodies within the cytoplasm. Coccus are spherical-shaped bacteria that often form clusters or chains. Bacillus are rod-like bacteria found in many environments. Prokaryotic cells vary greatly in shape and size. Prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO. Human red blood cells are about 7 micrometers in diameter. Mycoplasmas are among the smallest cells,
measuring about 0.3 micrometers in length. The cytoplasm is the main site where most cellular activities and chemical reactions occur. Cells contain many important organelles that help in their function and division.
Prokaryotic cells lack a membrane-bound nucleus. Eukaryotic cells contain a well-defined membrane-bound nucleus.
Human cheek cells have only a cell membrane as the outer boundary, unlike plant cells.
In 1855, Rudolf Virchow provided a crucial explanation. Schwann proposed that all plants and animals are made of cells and their products. An onion peel is prepared on a slide to observe plant cells under the microscope. He explained that cells divide to form new cells. He stated, "Omnis cellula e cellula," meaning all cells arise from pre-existing cells.
He observed that animal cells have a thin outer layer called the plasma membrane.
He studied plant tissues and compared them with animal tissues.
This formed the foundation of the cell theory.
He also noted that plant cells have a unique cell wall.
Schleiden and Schwann together formulated the cell theory.
However, the theory did not explain how new cells were formed.
All living organisms are composed of cells and products of cells. All cells arise from pre-existing cells.
Virchow modified the earlier hypothesis to complete the cell theory.
Let us observe cells as seen under a microscope and recall their basic structure.
The onion cell, a typical plant cell, has a distinct cell wall as its outer boundary and a cell membrane just inside it.
Human cheek cells are collected and prepared on a slide for microscopic observation.
Inside each cell is a dense nucleus, which contains chromosomes carrying genetic material, DNA. These structures are essential for understanding cell growth, reproduction, and the cell cycle. Cells can be broadly classified into prokaryotic and eukaryotic, based on the presence of a nucleus. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm fills the cell.
Eukaryotic cells contain membrane-bound organelles such as endoplasmic reticulum, Golgi complex, mitochondria.
Prokaryotic cells lack these membrane-bound organelles, making them structurally simpler than eukaryotic cells. Within the cell, ribosomes are found freely in the cytoplasm, where they synthesize proteins. Ribosomes are also present inside mitochondria, chloroplasts, and on rough endoplasmic reticulum. Animal cells contain a centrosome, a non-membrane-bound organelle that plays a key role in cell division. Cells show great diversity in size, shape, and function across living organisms. The centrosome helps organize spindle fibers during cell division.
Bacteria are larger than mycoplasmas, typically ranging from 3 to 5 micrometers in size.
The largest isolated single cell is the egg of an ostrich.
Nerve cells are among the longest cells in the human body. Cells vary greatly in shape, including disc-like, polygonal, columnar, cuboidal, thread-like, and irregular. These cells are generally smaller and multiply more rapidly than eukaryotic cells.